Drug nomenclature. Drug nomenclature. When a new drug is synthesized, it is first assigned a code of number, usually identified with its inventor, manufacturer, pharmaceutical company. If it is found promising after clinical evaluation, the manufacturer wants to put it in the market. The new drug is given a generic name to designate its pharmacological class, non-properties name. If the new drug gets official recognition by drug regulatory authority, its manufacturer give it a proprietary or trade name. Chemical name, IUPAC name, generally long and hard to remember. Generic, official approved word or trivial name. The chemical is entered in pharmacopoeias under this name. The chemical compound is known throughout the world by this generic name. Approved names are generally used by researchers and non-clinical teachers. Property, brand or trade name. One chemical compound can have several properties name. Even one properties name by may not contain single chemical. When manufacturers and clinicians prefer to use brand name. Example chemical name, generic name or trade name. 3,4-dihydroxyphenylethanomethylamine इपिनेफ्रिन है. ट्रेड नेम एड्रेनलिन है. एसिटिल सैलिसिलिक एसिड एस्प्रिन इकोस्प्रिन. Pharmacopoeia. It is a drug compendium consisting of officially recognized drug preparation or formulation. It gives the information on source, properties, purity, potency of recognized drug and these for their identity. A pharmacopoeia is the official publication of drug standard. It is revised regularly. The different pharmacopoeia are IP, the International or Indian Pharmacopoeia, USP, the United States Pharmacopoeia, BP, the British Pharmacopoeia, BCP, the British Pharmaceutical Codex, EP, European Pharmacopoeia, IVP, the Indian Veterinary Pharmacopoeia, BVETC, the British Veterinary Codex, NF, the National Formulary USA, NF, the National Formulary of India, BNF, the British National Formulary, ADR, the Accepted Dental Remedies, NB. The abbreviation of the pharmacopoeia is mentioned after the name of the drug. For example, benzoic IP, the tincture of benzoic corresponding to the standard tincture of benzoin mentioned in the Indian Pharmacopoeia. Sources of drug. Drug from plant sources. The ancient or original sources of drug are the plant collectively known as medicinal plant. All parts of the medicinal plant have therapeutic value. Root, sarpaganda, rhizome, ginger, haldi, bark, cinchona, catechu, acacia, leaves, atropine, cocaine, physoxygamine, flower, digitalis, chrysanthemum, fruits, papaya, anise seeds, nux vomica, kali mirchi, methi. Drug from animal sources. Hormone, oxytocin, insulin, thyroxine, gonadotropin, vitamin, cord of shark, liver oil, rich source of vitamin A and D, antisera, antisnake venom, canine distemper, antiserum etc. Other heparin, liver extract, immunoglobulins, blood plasma. Drug from microbial sources. Fungi, actinomycetes, source of antibiotic, penicillin, streptomycin, and gentamicin, neomycin. Virus, bacteria, preparation of vaccine, yeast, dried yeast as source of vitamin B complex. Drug from mineral sources, inorganic salt. antacid magnesium oxide, sodium bicarbonate, purgative magnesium sulfate, expectorant potassium iodide, diuretic potassium nitrite, hematinic ferrous sulfate, hypothyroidism iodine, mineral oil, liquid paraffin, laxative effect, long-term administration interference with vitamin A, D, calcium and phosphorus absorption. Synthetic drug. Majority of the current day drugs are from synthetic sources. Example are antipyretics, barbiturates, tranquilizer, anti-inflammatory drug, anesthesia, antiseptic, antiprotozoal, antihistaminic, etc. Semi-synthetic drug example are agonist and antagonist of morphine, dihydrotriptomycin from streptomycin, semi-synthetic penicillin from penicillin. Gene therapy. Gene therapy. It means prevention or treatment of disease through manipulation of gene function. This is insertion of specific genes, therapeutic genes exogenous into the animal cell. The concept of gene therapy has its origin from the fact that manipulation of gene expression could change the function of abnormal genes or supplementation of a non-functional gene or suppression of an abnormal gene. Gene therapy refers to introduction of functional genetic material into target cell to replace or supplement defective genes or to modify target cells so as to achieve therapeutic goal. In contrast to all other drugs, the kind of therapy can impart new functions to a cell. Gene therapy holds a great promise for curing a number of diseases which at present can at best be only palliated or controlled. Gene defect result in failure to synthesize a functional protein or in the synthesis of a dysfunctional protein, equipping the cell, especially the one which physiologically express it, with a normal copy of the defective gene would overcome the deficiency at the site where it is needed on a long-term, maybe permanent basis. Recombinant DNA technology from the basis of synthesis of therapeutic gene. Technique of gene delivery into host. Technique of gene delivery into host. It is highly complicated and different from that of conventional drug delivery system. The technique involves inserting a therapeutic gene first into a vector. The vector may be either viral, retro, or adenoviral, and non-viral vector plasmid DNA, liposomes, microsomes. A vector with a gene is then introduced into the patient through either in vivo or ex vivo means. In vivo gene transfer. The vector, usually a retrovirus carrying the gene, is injected systemically or directly into the concerned organ. Ex vivo gene transfer. The patient tissue cell, blood or bone marrow are isolated and maintained in tissue culture. These are then transferred with vector carrying the relevant gene and injected back into the patient. Application of gene therapy. Cystic fibrosis, severe combined immunodeficiency disease, growth hormone deficiency, Parkinsonism, HIV infection, Alzheimer's disease, Huntington chorea, cancer, hypertension, hemophilia, insulin dependent diabetes, etc. Biopharmaceutical. Biopharmaceutical. These are therapeutic agents produced through biotechnological means but not by conventional laboratory chemical synthesis. The principle of biopharmaceutical and the process of their development have origin from the advancement in the knowledge of molecular cell biology and biotechnology. Therefore, biopharmaceutical, popularly known as designer protein, are the promising therapeutic tools of the future. Example are functional human peptide ADH, oxytocin, GnRH, ACTH, TSH, TRH, calcitonin, insulin, somatostatin, growth hormone, cyclosporin, etc. Enzyme peptide, streptokinase, asparaginase, DNase, erythropoietin, clotting factor, interferons, monoclonal antibodies, vaccine, etc. Active principle of medicinal plant. The mechanical value of plant or crude preparation of medicinal plant is due to presence of a variety of pharmacologically active principles such as alkaloid, glycosides, oil resin, oleoresin, gum, saponin, tannin, etc. Alkaloid, basic nitrogenous substance, insoluble in water, less soluble in alcohol, soluble in ether, chloroform, and oil. From water-soluble crystalline salt with acid, alkaloid consisting of oxygen or solid, atropine, reserpine, emethine, morphine, stretching, quinine, etc. Alkaloid without oxygen or liquid, nicotine, pilocarpine, lobeline, etc. Mostly derived from plant, exception epinephrine, obtained from adrenal medulla. Alkaloid atropine source action. Alkaloid source और एक्शन एट्रोपिन एट्रोपोबेलारेना एंटीकोलिनर्जिक एक्शन है अरेकोलोनिक अरेका कैचक्यू कोलिनर्जिक इफेक्ट है कैफीन कॉफ़ी या अरेबिका कॉर्टिकल या सीवीएस स्टिमुलेंट कोकेन इथ्रोजाइलोन कोका लोकल एनेस्थेसिया सीएनएस स्टिमुलेंट इमेटिन सिबेलिस इपेकेचुआहा रिफ्लेक्स morphine, peperosomniferum, narcotics, analgesic, nicotine, nicotiana tobacum, ganglionic stimulant blocker, physostigmine, physostigma venosum, anti reserpine, rifolfia serpentina, antihypertensive, strechine, stretchenox vomica, स्ट्राइक्नोज नक्सोमिका स्पाइनल स्टिमुलेंट या कॉन्वोल्यूजेंट ग्लाइकोसाइड्स कंपाउंड्स containing a sugar glycone and a non-sugar a glycone or genin part joined together through an ester linkage, so these are sugar ester. The pharmacological action resides in the a glycone या genin. Glycone part determines solubility, tissue permeability, and duration of action of a glycone. Glycoside do not form salt with acid on acid, alkali, or enzyme hydrolysis. The glycoside break into two parts. Example: glycone and aglycone. Example category: glycoside source: cardiac glycoside. Cardiac glycoside. Digitoxin, detoxin, digoxin, gitaline, strophanthine, quabain. Source: digitalis lantana और purpurea leaves से, strophanthus gratus seed से, यूरिजेनिया मेट्रिमा bulb से. Cyanogenic glycosides: एमाइगडालिन जो मिलता है प्रूनस एमिगडालस, धूरिन, सोरगम वल्गेरे, लिनामेरिन, लिनम उशिटेसिमम. मिसcellaneous glycosides. Miscellaneous glycoside: मैग्नेफेरिन हिपेटोप्रोटेक्टिव या हेमेटोप्रोटेक्टिव और एंटीऑक्सीडेंट. स्वेटियामारिन मैन्फीफेरा इंडिका लीव्स और फ्रूट से. स्वेटियामारिन कार्डियोटोनिक या हेपेटोप्रोटेक्टिव. स्वेटिया चिरकाराटा स्टेम लीव. ऑयल: these are of two types: fixed oil and volatile oil. Fixed oil: these are glyceraldehyde of oleic, palmitic, and stearic acid. Many fixed oil have food value. Cooking oil: example corn, groundnut, sunflower, mustard, soybean, coconut, palm oil, etc. Cooking oil are pharmacologically insert and serve as vehicle for fat soluble vitamin Some other have pharmacological action. Example: fixed oil source or pharmacological action. Castor oil, resin, communis, purgatives, linseed, sour cell, linum, उस्टेसिनम डेमुल्शन वेहिकल पurgative के फॉर्म में क्रोटन ऑयल क्रोटन टिग्लियम ड्रेस्टिक purgative में volatile oil also known as aromatic essential ethereal or flavoring oil. These have no food value. These are volatile and emit characteristic odor while evaporation. Most of these have medicinal value. Example: volatile oil source pharmacological action. Eucalyptus oil, Eucalyptus globulus, expectorant, rubefacient. Ginger oil, Ginger officinale, stomachic, carminative. Terpene oil, Citrus deodora, counter irritant, astringent. Clove oil, Eugenia caryophyllus, analgesic, antiseptic. Peppermint oil, Mentha piperata, antiseptic, antiemetic. Asafoetida oil, Ferula foetida, carminative, antihelminthic. Resin. These are brittle, amorphous compound formed from oxidation or polymerization of terpene compounds of volatile oil. These are insoluble in water, soluble in alcohol and other organic solvent. From soap with algae. Example: resin, colophonium. Resin source pharmacological action. Colophonium, residual left after distillation of crude terpenes. Antiseptic, septic astringents. Podophylline, dried rhizome and root of Podophyllum immodi. Purgative, sialic, cholicogens, antimycotic, anti-cancer. Oleoresin. These are mixture of volatile oil, gum and resin. Example: oleoresin source pharmacological action. Asafoetida, secretion from root of Ferula foetida, carminative, antispasmodic. Balsams are also considered as oleoresin. These contain an aromatic acid, resin and volatile oil. Example: balsam of tolu, secretion from trunk of Myroxylon toluferum. Pharmacological action: expectorant, antiseptic. Balsam of Peru, secretion from trunk of Myroxylin peroic, antiseptic, acaridic. Gum. These are polysaccharide secretory product of plant capable for forming thick mucilaginous colloids when mixed with water. Gum are pharmacological inert with no systemic effect but exert demulcent action on surface and are mainly used as suspending or emulsifying agent in pharmacy. Gum source pharmacological action: agar, colloidal carbohydrate from seaweeds, bulk purgative. Gum acacia, senegal, demulcent, emulsifier. Gum arabica, acacia arabica, demulcent, emulsifier. Gum tragacanth, astragalus, gummifier, demulcent, emulsifier. Saponins. These are non-nitrogenous substances soluble in water which form or froth when shaken with water. Saponin upon hydrolysis split into a sugar and a non-sugar saponin, hence considered as a subclass of glycoside. Saponin causes hemolysis of blood. Example: Quarelis, Seneca, etc. Tannin. These are water-soluble non-nitrogenous plant constituent having characteristic astringent action precipitation of for a protein upon mucous membrane. These exert a protective action on the mucosa GI against irritant. Tannin also inactivate alkaloidal poison. Tannin source pharmacological action. Catechol Ancaria gambier astringent precipitate alkaloids. Kino Pterocarpus marsupium astringent precipitate alkaloid. Giela Quercus infectoria astringent antiseptic. Antiseptic. Chapter complete.